As we begin the Science Symposium, if this is your first time with EHE 360, I want to introduce David Casimir. He's our expert moderator for the event. David serves on the EHE Foundation Board of Directors, and his professional expertise as an intellectual property attorney and scientist make him a perfect contributor to this forum. So David, welcome. Thank you for your service. I'm going to hand the meeting over to you now to introduce our first session and take over the day.
Excellent. Thank you for having me again this year. Our first session is going to be on the topic of novel approaches to target EHE fusions. We just learned from Dr. Chen that EHE is linked to 2 specific gene fusions, and that allows scientists to look at those fusions as ways to potentially target the disease and identify treatments and cures. And our first presenter today will be Dr. Harvey from the Peter MacCallum Cancer Center in Australia at Monash University. And he's going to present on real-time investigation of EHE fusion protein behavior. Dr. Harvey, please take it away.
Okay, thank you very much, David. And thank you for the invitation. It's great to be here speaking to you from Australia. So as David mentioned, I'm going to tell you about some ongoing studies in the lab where we're looking at studying EHE fusion proteins in real time using high resolution microscopy. But first, a bit of background. So my foray into the EHE world started around 20 years ago when I was a postdoc at Mass General Hospital in the early 2000s. At the time, we were looking to identify new cancer genes, and we did that by using a large-scale screen in the fruit fly Drosophila. And so we aimed to identify genes or mutants that had these large overgrowths in the head. And then we went on and characterized the human orthologs of those and tried to understand whether they were mutated in cancers. And so this was a very successful screen that was conducted in Ishwa's lab as well as some other labs at around the same time. One of the major things that came out of that screen were the founding members of a brand new signaling pathway, which we now know is called the Hippo pathway. And you can see here a fly on the left that has a beautiful ordered array of ommatidia in its eye. And then on the right, we have a Hippo mutant, which has all of this excess tissue growth because the cells grow and divide in an uncontrolled fashion, and then they don't die when they should, similar to what happens in a cancer. And we also found that some of these founding members were actually mutated in cancer. So that was the first evidence that this pathway is important for human cancer. And I've really studied that signaling pathway in my lab ever since. And then we got into EHE in the early 2011, around 2011, when Munir Tanas in Brian Rubin's lab, as well as Christina Antonescu's lab, discovered the first EHE fusion TAS-CANTA. And, you know, at the time, we knew that YAP and TAS were oncogenes, as was introduced in the first presentation by Dr. Chen. But unlike other oncogenes in cancer, like RAS and RAF and PI3 kinase, we didn't really have many obvious activating mutations in YAP and TAS in cancer. And so we were really intrigued when these papers came out that showed that TAS initially and then subsequently YAP were fused to different other genes in the context of cancer. So this was, you know, a potential mechanism by which these oncogenes are hyperactivated. And so we were really interested in that. So we think of the Hippo pathway like a brake. You know, if you use a car analogy, we think the Hippo pathway is the brake, and then the accelerator or the gas is the oncogene, which is YAP or TAS. And in a normal system, that is regulated. So you can stop and start that car when you need to. But what happens in EHE is you have this hybrid protein. So now you have the accelerator portion of YAP, but you have the brake to the Hippo pathway is essentially cut. So So you've got this oncogene, which is now unregulated, and it's driving growth and proliferation when it normally shouldn't. And so we started our work on EHE in sort of the mid 2015s, and we did that in collaboration with two wonderful scientists. First of all, Munir Tanas, who, as I mentioned, was one of the discoverers of the TAS-CAMTA fusion protein in EHE, and Anne-Claude Gingras, who's a wonderful biochemist based in Canada. And so we had a paper published in 2021 in eLife where we did some molecular studies. You know, we're really intrigued by the fact that you had these two oncogenes at the base of the hippo pathway that could be fused to two different genes. So we wanted to know, are they driving the same sorts of genes to transform cells and cause cancer, or do they operate by different mechanisms? And what are the proteins that they work with to drive transcription and cell proliferation? And so we did a range of molecular studies aimed at answering those questions. What are their target genes? What do they bind to in the genome? We wanted to understand whether they drive proliferation via TEADs, these key DNA binding proteins in the hippo pathway. And then we also wanted to understand what are the different transcription regulators that these proteins recruit to the DNA to change gene expression, because maybe that will give us some other therapeutic opportunities. And other scientists around the world have done similar studies on TAS-CAMTA, YAP, TFE3, and other fusion proteins involving YAP and TAS in different sarcomas and other cancers. And then more recently, we've been taking a very different approach to try and gain new information into EHE fusion proteins as well as the parent proteins by using high-resolution microscopy. And we've done that with the generous support of EHE Foundation in Australia, as well as the EHE charity in the UK over the past couple of years. And so what we've tried to do is to understand the molecular mobility. So how do these proteins navigate throughout the cell and throughout the nucleus to find their target genes? How do they bind DNA? What are the molecular properties of that interaction? And then we also wanted to see how some of these emerging potential EHE therapies impact these fusion proteins using live microscopy. And so the data is actually really stunning to look at. So first of all, we studied these fusion proteins, but we also studied the parent proteins. By that, I mean YAP as well as TAS, and we're also looking at TFE3 alone and CAMTA alone. So what we can do is tag YAP with a little protein called halo, and then we can make it glow at single molecule resolution by adding a little ligand that fluoresces under the microscope. And so the raw data looks something like this. Here we're looking at a single cell. In gray is the DNA, and around that is the cytoplasm. And what you can see is every one of those little spots is an individual YAP protein. And normally in microscopy, we study things on a population level. And it's only relatively recently that we've been able to use new technologies to investigate protein at single molecule resolution. So we can gain a lot of information from this. First of all, you'll see that it's incredibly dynamic. Proteins are moving around very, very rapidly throughout the nucleus and the cytoplasm. And occasionally they're sitting still, and that's when they're binding to DNA. The data is very quantitative, so we can use computational algorithms to track the behavior of every one of those individual proteins. And you'll see already, just from looking at the tracked data, that you see some proteins or some little spots where the protein seems to be very immobile. That's when it's binding DNA. And then you'll see other traces where you can see the protein randomly moving throughout the nucleus. So two broad types of behavior, mobile and immobile. And we can track all of that behavior because we're taking very rapid acquisitions. So we take around 50 frames per second to capture all of that data. Okay, so that's the parent protein. How does a fusion protein look? And so for this, we're looking at YAP-TFE3, which is found in around 10% of EHE cases. So first of all, again, we're looking at a single cell. In gray is the nucleus. And this is YAP-TFE3 protein at single molecule resolution. So you can see already compared to YAP, it's more nuclear, which is something that we already knew from previous studies. But you can see that it's also less mobile. So there seems to be more YAP-TFE3 sitting down on the genome rather than rapidly diffusing around, although there is still some of that behavior. When we quantify that, or we also wanted to look at the beauty of this system is it's very amenable to manipulation. First of all, genetic manipulation, but also chemical manipulation. So in this scenario, what we did is rather than looking at YAP-TFE3, we mutated a single amino acid in YAP that prevents its ability to bind to TEADs, because we know that that's how YAP contacts the genome. And then we looked at what that did to fusion protein behavior. And you can see here that it really transforms its behavior quite significantly. We're now starting to see a lot more protein in the cytoplasm outside the nucleus. We're starting to see much more rapid behavior, but we do still see some immobile events. And when we can quantify all of that behavior, and that really gives us a beautiful quantitative picture of what's going on. So if we first focus on the blue trace here, we see two broad types of behavior, which we classify as immobile, based on the amount of these of movement of these molecules in a given time, and then mobile behavior. And you can see for YAP-TFE3, it's mostly immobile, but then we also see some mobile behavior. Now, when we mutate just a single amino acid that's required for this protein to contact TEADs, we see it completely transforms its behavior. And the behavior now shifts largely from the immobile pool into the mobile pool. And that's because it's not being able to contact DNA as well as YAP-TFE3. And if you look at the tracks of these side by side, that really beautifully illustrates that. You can see YAP-TFE3 has many more spots, and these are the immobile DNA binding events. And then when we mutate a single amino acid, we're now seeing far more random diffusion of this protein throughout the genome because it can't bind to TEADs. Okay, and just to hammer home the point of how important TEADs are for YAP-TFE3 to bind to the genome, we also overexpress TEADs, so put more than what the cell would normally make. And then we did single molecule tracking again. And now you can see that the behavior changes yet again. We're seeing that almost exclusively, we're seeing YAP-TFE3 individual proteins being bound to DNA and exhibiting this immobile behavior. And you get these really beautiful single molecule traces from this experiment. Okay, so that's looking at the whole population of proteins in a nucleus by imaging 50 acquisitions per second. When we want to look at just that immobile behavior, we take 2 acquisitions per second. And what happens there is all of the really fast-moving immobile molecules blur out of focus, and we look only at the immobile population, which equates to the DNA binding population. Okay, so as I mentioned before, we did this for YAP-TFE3 fusions, but we also did it for YAP and TEAD, which are important in EHE, but also in other cancers. And first of all, this is the DNA binding behavior of YAP-TFE3 in the blue, then this mutant that can't bind to the TEAD proteins in the magenta trace. And what we see is something that is seen for many transcription factors, and that is that they bind to the DNA on a broad set of scales or broad time scales. So this is the log scale. You can see that we've got events that we're recording for 1 second or a couple of seconds. But it's really a broad continuum right out to around a couple of minutes. And the other thing that we see is that the blue trace, this is just the raw data, is exhibiting longer DNA binding times than the protein that can't contact the TEADs. We can then extract from that DNA binding times for these different proteins. And so what we've done here is we've looked at TEAD. This is the DNA binding protein of the hippo pathway. And we can see that this binds to the genome on average for around 40 seconds. So this is really important information, foundational information that we can use to compare and contrast to a lot of different settings, including adding chemical inhibitors or genetic perturbations, etc. YAP in green binds to the genome on average around 20 seconds, so around half of the time. So what that tells us is that YAP and TEAD aren't always together. It tells us that the complex of these two proteins must be dynamic, or otherwise they would show the same DNA binding time. So what about the fusion protein? Okay, so here we're looking at YAP TFE3, and I've drawn it in the model as contacting DNA through TEAD. So we went into this with a completely open mindset. We didn't know whether YAP TFE3 would bind to genome longer or shorter. But what we found is it actually binds to the genome a lot longer than YAP itself. So YAP's around 20 seconds. YAP TFE3 on average was almost a minute at around 54 seconds. So almost 3 times longer than YAP. So it's forming more stable complexes with DNA. And it was even longer than TEAD itself. Okay, so what about the mutant that can't bind to TEAD? So this is the YAP S94A TFE3. So this has a dramatically reduced DNA binding time of around 20 seconds. Now, what we think is happening here is that this protein can no longer bind to TEADs to contact DNA, but we know that TFE3 still has DNA binding capacity from many different studies, including the work that we did with Munir and Claude. So we think what's happening here is TFE3 is still anchoring this protein to the genome to some respect, but that is doing so at a much shorter timescale than YAP TFE3 itself. Okay, so I'm going to stop there and just give you a summary. So first of all, we've used single molecule tracking microscopy to understand and quantify the nuclear behavior of YAP, of TEAD, and YAP TFE3, as well as the times that these proteins bind to DNA. And we've started to probe the importance of cofactors such as TEAD in this process. And we've shown that TEAD already is really important for YAP TFE3 and in terms of its nuclear mobility as well as its DNA binding. And this and some other results were published by our team last year. So what about what's next? So we're really interested and have a lot of ongoing work to expand these studies to TAS-CAMTA, the major fusion in EHE, as well as the parent proteins TAS-CAMTA and TFE3, and then also explore how drugs, such as the ones that Dr. Chen mentioned, the TEAD inhibitors, how they are impacting the nuclear mobility and the DNA binding capacity of the fusion proteins, as well as their parent proteins. Okay, I'm going to stop there, and I just want to acknowledge all the fantastic people in my team that have been involved in this work, in particular Vashini Mohan, who's directly supported by the EHE Foundations, as well as Ben Kroger, Sam Manning, and Gigi Deng. Our collaborators, Munir and Claude and Matt, and I also want to give a big shout out to the EHE community, and in particular, thank Jono and Di from the EHE Foundation Australia, as well as Hugh Leonard from the EHE charity in the UK, and Guy Weinberg, who many of you will know, who established this fantastic meeting in Telluride, where many of us get to come together and share our latest science. Thank you.
Thank you much, Dr. Harvey, and we'll have you go off camera, but be back in a little bit for a group Q&A. And just for everyone online, we have been seeing your questions, and we will get those answered in the group Q&A. But now we're going to turn over to our second presenter in session one, which is Dr. Bauer from the University of Tubingen and University Hospital, who's going to talk to us about immunopeptidome-guided target discovery for T-cell-based immunotherapy. Please take it away.
Thank you very much for the kind introduction. I hope you can see the slides. Thank you for inviting me here today. So I'm going to present my project and also the technology behind it for how to identify via immunopeptidomics tumor-associated antigens that can be used for T-cell-based immunotherapy. And this work is now recently funded by the EHE. And so I've first given an overview of the technology and what we can do with it right now, and then a little bit about the EHE project that is now starting. So just so if not everybody knows where Tübingen is, the university clinic where I'm working, it's in the southern part of Germany, right here. It's a very beautiful city. And in the University Hospital in Tübingen, we are working in the department for peptide-based immunotherapy, which is headed by Professor Juliane Walz. And we have three working groups within this department: the research unit, which is headed by me together with my colleague, Dr. Annika Nelde, where we are doing our mass spectrometry-based immunopeptidomics studies and characterize T-cell epitopes. We then also have a unit where we can produce peptides in GMP quality, which we use for our vaccine platform. And then a clinical unit, the co-head under Juliane Walz and Professor Helmut Sali, where we can apply these in clinical studies directly in patients. With this pipeline, we are characterizing and developing T-cell-based immunotherapies. And just giving a little bit background about this, when we talk about the antigens we are characterizing with immunopeptidomics, we are talking about small protein fragments on the cell surface of cells, and in oncology, of course, on cancer cells. These HLA molecules present these peptides on all living cells with a nucleus in the human body. And it is possible to identify cancer-associated antigens that are presented due to dysregulation of the cancer, of the metabolism, or through gene mutations. There can arise differences in these peptides, and we can measure this via mass spectrometry. A very common approach how to also target these antigens is to do algorithm-based prediction, so doing a sequencing of the tumor and then predicting what neoantigens might be presented and then targeting these. However, with this approach, you also miss a lot of targets, or a lot of the targets that you are targeting are actually not presented on the living tumor. So what we are doing is directly analyzing what the tumor is presenting. So we are doing an affinity purification of the naturally presented peptides from tumor tissue, and then measure them on an online coupled LC-MS, so mass spectrometer, to sequence the peptides that are presented. This gives us, in the end, mass spectra, which we can sequence and then directly translate what peptides are really presented by the tumors and can be targeted. This methodology was spearheaded by Professor Hans-Georg Ramsey, who is now retired, and we are continuing this work in the department right now. So this is the technology we are using. Now, giving an example where we are applying this methodology, it is another tumor entity which is characterized by a fusion gene, the fibrolamellar carcinoma, or short FLC. Maybe you have heard of it. In this project, which was published in 2022, we were working on the question, is this fusion gene, which is occurring in 100% of the FLC patients, is it naturally presented by these HLA molecules on the tumor cell surface? And can we use it as a target? We did then immunopeptidomic study from these samples, and could very luckily identify naturally presented antigens directly from the fusion region, which is then a specific neoantigen that was presented by these cells. In a single patient study, we formulated a vaccine containing 4 different peptides, all covering the same fusion region of the patient. And after 2 vaccinations, we could see a very strong induction of T-cell responses in this patient. Patient was at a very dire clinical setting, and after the vaccination, there were no further relapses anymore. So now, even 5 years later, we are still monitoring the T-cell response of the patient, and she's still in complete recovery. So we could show that this approach really works here. From this, we have now started 2 clinical studies in Tubingen evaluating this vaccine, focusing on solely the fusion. And the first clinical study is combining it with a checkpoint inhibitor, and the second clinical study is in the adjuvant setting, and both are currently recruiting and running. And we are looking forward to the data that we are generating here. To give you a little bit overview of how the vaccine is formulated. So this is now the example. This was an example of what antigens we can target, and especially fusion antigen. And we are formulating these vaccines by combining our novel adjuvant, the XS15, which is a water-soluble TLR1/2 ligand, together with peptides in a water emulsion in montanide. So we mix water and the oily liquid montanide, which is also an adjuvant, and generate a very persistent emulsion, which we then inject in the belly area in the subcutaneous fatty tissue. This gives us the unique opportunity to having a very strong persistence of the peptides, because peptides in general are rather unstable, but if you formulate them like we are doing it, they are very persistent at the vaccination site. And this has 2 big advantages. The first one is they stay there for a very long time, and this leads to a very strong induction of T-cell responses. The other good thing is that we have a very good safety profile. The peptides are not going systemic. They are only reacting at the vaccination site, which we also could show here in the PET trace, a very strong activation here. In this local granuloma formation, so there's a local swelling and a local inflammation process, which is intended. But it's not going systemic, which is a big advantage compared to mRNA vaccines, where you have a lot of side effects like fever or other stuff if you go into very high antigen amounts of antigens. And we could show that we could induce very strong T-cell responses with this, also stronger responses compared to mRNA vaccines. This is here for our SARS-CoV-2 vaccine compared in the figure. And we have published this now in several studies in healthy volunteers and also in cancer patients. And we've now vaccinated over 100 people with this formulation of an antigen vaccine. Going now one step closer to the project, still staying in FLC. However, what we observed is that having a single targeting vaccine might not be the best case, and we are right now developing this further. Here showing one patient case where the patient had several metastases already at initial disease, and was treated over several months until we could vaccinate with the fusion antigenic targeting vaccine cocktail. After this, we could see, as in the first patient that I showed, an induction of T cells against this antigen. However, it was a minor reaction by the T cells. So shown here on the scale, the response was in the 1,000 spots, and here we are around at 50, so it was a borderline activation. Even though we vaccinated several times, even after the 4th booster, it only went up a little. So we were thinking about how can we improve this cancer vaccine even further. And this is another perfect example of why immunopeptidomics is a wonderful technique to study antigens and tumors, because we are not only we don't have to rely on only the neoantigens that we can identify with this technology. We can identify everything which is naturally presented by the tumor, and analyzing this, and that is also what we did over the last decade. We have measured hundreds of malignant samples and also corresponding benign samples, and we can identify from a single tumor thousands of antigens naturally presented, and using this comparative algorithm to compare in our database what did we find on tumor and what did we find on on benign samples, we can identify high-frequent tumor-associated antigens that are even unmutated. And this is a broad spectrum of antigens that we can also use for our vaccines. Here, very broad overview of the antigens that we can identify with this. I won't go into the detail here, but the first category, the mutated antigens, is of course what the fusion genes are falling into, and then the second big category is the non-mutated antigens that are presented due to deregulation of the tumor, of the metabolism, or of the genetic landscape of the tumor. And we can really identify antigens with high frequency of up to 75% in the whole population of tumors that we analyzed. And we never before found them on any benign tissue, even though we measured hundreds of them via immunopeptidomics. So coming back to the patient that I just showed, looking at the whole immunopeptidome, we found roughly 9,000 different antigens, the peptides on the tumor tissue. And then comparing this to the database that I just was explaining, we could identify antigens within this immunopeptidome here in an FLC patient that we could find in various different cancer antigens, cancer entities as good cancer antigens. Here just showing the top 7 that we selected, which was also very abundant in glioblastoma, melanoma, hepatocellular carcinoma, and other entities. So we formulated a new vaccine having the fusion gene and also these non-mutated antigens, the top 7 that we selected, and vaccinated the same patient. And with this revaccination, we could see a very strong induction of new T-cell responses against the non-mutated antigens, but also accompanied by very strong boost of the fusion gene response, formulating here a better vaccine. So with this, with the technology now described and what we can do with it on other cancer entities, I want to now switch over to the project that we are now starting in Tubingen, so, on EHE. So I don't think I have to say much of EHE. The introduction there was already very, very good. So it's a rare vascular sarcoma, and it's really defined by these fusions, the TAS-CAMTA or the YEP1 TFE fusion. And we are in this project, of course, wanting to look if naturally presented peptides from these fusions arise in EHE samples. However, we also want to look at the unmutated and the whole immunopeptidome of what we can identify to try to characterize useful antigens which could be applied for clinical translation to target EHE. So the project is called Immunopeptidome-guided target discovery for T-cell-based immunotherapy in EHE, and this is now funded by the EHE, and I'm very glad to receiving this opportunity. We are planning to measure a cohort of 10 to 20 patient samples, so tumor samples, which we are right now already starting to collect. I'm collaborating on this with the lab of Professor Gujal from the Fred Hutch, and we're currently also reaching out to the different biobanks, the US EHE biobank and the UK biobank we already wrote to, and this is now in the work, receiving the samples here. And we have set up also a sample collection in Tubingen, where we have a national sarcoma center, and we have already the first patient who might get resected in the next weeks, where we might be able to collect the first tumor tissue here in Tubingen. So when we have the first tissue in the lab, we will start then analyzing the tumors. We will do immunopeptidomics of the tumors, the tumor tissue. We will do HLA typing if we can from the tumor or from a blood sample that we can collect to have a better characterization of the immunopeptidome of the patient. And if available, we will use the sequencing information of the fusion or other multi-omic data to integrate our data sets and selecting and characterizing the immunopeptidome of these patients. So isolating, identifying naturally presented peptides from these tumors via mass spectrometry, and then doing our algorithm of selecting the malignant associated antigens from these, hopefully identifying fusion gene-specific ligands from these tumors and combining this with other high tumor associated.
Looks like we have a technical glitch. We'll give Dr. Bauer a minute to see if he can log back on. As we're waiting, let's have Dr. Harvey come back on, and we'll start the Q&A session just in case we're not able to continue that presentation. We'll make sure the full slides are available later on. And I'd also like to introduce from our team, Dr. Rishton, a new, relatively new addition to the EHE Foundation and an absolute superstar. We are all thrilled that she's been able to come on board. Her PhD research work was in fusion-driven sarcomas. She has expertise in the Hippo pathway and preclinical models for testing these types of things. So an absolute brilliant scientist. We couldn't be happier to bring her on board. And she's going to be helping moderate these Q&A sessions with me.
And Jen's
we got Dr. Bauer back.
I'm so sorry.
That's
I
okay.
think,
No problem, that happens. Do you have a few more slides you'd like to finish off?
I don't know where exactly I was switched off.
You
Sorry.
broke off on the slide showing, yeah, and I can help you back it through there. Right there.
Right
And
there.
you had actually gotten all the way to the last subpart of that one.
Okay, so, okay, then, yeah, actually, then I was already nearly at the end. I just wanted to thank everybody that was working on it. I'm so sorry, my internet connection was, I don't know, completely skipped working here. So I want to thank everybody involved in this project. I want to thank all the collaborators, all the people in the lab from the Department of Peptide-Based Immunotherapy. I want to thank the EHE Foundation to funding me and helping me setting up my lab here, and all the other funding agencies. And of course, I want to thank all the patients and families that make this kind of research possible. Yeah.
All right.
And
Yeah.
sorry again,
It's okay. I'll have the first question for you. And it's, so from what I understood from your presentation, the EHE tumor cells, for whatever reason, are going to look different than the non-tumor cells. And we expect that there to be things on the surface of the cell that you will be able to potentially identify that will say this is an EHE tumor cell and not a normal cell, and that those then could be targets for vaccines or other approaches. Can you give a little more detail on if we identify some unique targets that are EHE specific, what would be the next steps in development of a treatment? What would that look like?
Yeah, so very nicely put together what we are planning to do as isolating and characterizing these antigens from the point that we have valid antigens where we know these are presented naturally and they are immunogenic. We have translated now these kinds of peptide vaccines in several different entities. We have a clinical trial running in CLL, AML. We're starting one in prostate cancer and head and neck cancer right now. And we have the fibrolamellar carcinoma samples ongoing. Because it's kind of a bit with the mRNA COVID vaccines, the whole setup that we have is up and running and it's all approved. We have the vaccine formulation, we have the adjuvant. The only thing that we have to change out are the antigens that are cancer specific. Sadly, there are little antigens that are broadly presented in a lot of different tumors. So we always have to, in each entity, look especially what is there, the specific antigens there. But if we really can identify tumor-specific antigens, the next steps would be quite fast to translate this maybe in a clinical study or in a first individual healing attempt. The German medical laws, they're quite open. Like if you have a treatment for a patient where no other options are left, if you can manage to produce the vaccine under GMP conditions, you can quite fastly translate into the clinics. And this really is facilitated by this department of where we have the production, the research, and also the clinical part within one department in Tubingen. Yeah.
Very exciting. Christy, any questions for the group?
I do. So, Dr. Harvey, this is for you. You talked a lot about the relative mobility of all of these different proteins, both the parent proteins, the YAP and TAS proteins, but also the fusions that they have with them. And I understand that the goal of TEAD inhibitors in general is to kind of change the way that those proteins interact with each other. Can you imagine or think of a way in the drug development pipeline that we might be trying to specifically target how the proteins are moving in and out and around the cell? Or are we really just looking at changing the way that the proteins interact with each other.
Yeah, I think, Christy, the main thing is we want to try and prevent that interaction of YAP and TAS with TEAD, or the fusion proteins with TEAD, because we know that that is driving transcription, and we also know that that's driving transcription of genes that are promoting the tumor. So that's really the primary goal, is to try and block that interaction, and potentially even, TEAD can be a transcription repressor on its own when it can't bind to YAP and TAS, or the fusion proteins. So we're also trying to kind of flip that switch and make TEAD, instead of driving gene expression, blocking gene expression.
Great. Thank you so much.
Yeah, and this is a question from one of our audience members. I think this is probably directed at Dr. Bauer. There's a question about RNA vaccines as a potential approach for EHE, and I guess maybe opening the question up more broadly, because you had done some comparisons in your slides with your prior model. What are your thoughts on RNA vaccines as an approach versus peptides, and the different vaccine approaches when you do identify a novel target?
Yeah, thank you for the question. So in general, of course, I'm a little bit biased, but however, I think that peptide vaccines have some unique advantages compared to mRNA vaccines. However, both are viable options. Like in the end, if you have identified suitable antigens, there are many different ways how to apply immunotherapy. There are also still around. There are different ways to do it, and I think yes, the fusion might be a great antigen. The question is really, is it presented by HLA or not? If it's not presented by the tumors and we cannot identify it in immunopeptidomics, then it's not a useful target. And this sometimes happens because, of course, the tumor wants to evade the immune system. This is why also we think with this broad approach of not only focusing on the fusion, but having the complete immunopeptidome analyzed and looking for different options might be the best way to go. But yeah, mRNA vaccines are also a possible way to target these fusions.
Excellent, thank you.